is it? It's a solid carrier family 18-member A1, and it codes for vesicular monoamine transporter VMAT1, a protein that packs cytosolic monoamines into presynaptic vesicles. And I think it'll be great because Rebecca will sort of talk about the synapse after methamphetamine. So basically, we have dopamine. Dopamine signaling is important. It's released presynaptically, and then when it's in the synaptic cleft, it gets reabsorbed usually in the extrasynaptic space, and it's all like in the cytoplasm. And molecules in the cytoplasm is usually not good because it's too exciting for the cell, and when the cell is too excited, it dies. So all these molecules have to be sort of recycled into little packets, and that's what actually this protein does, the protein that is produced by this gene that we identified. So we thought, okay, before we chase this gene, is that relevant? And relevant as in, is it relevant to humans? Because there are genes that humans have that animals don't have, and vice versa. And it is definitely expressed in the human brain. So the thicker the band, more expression. And also it turned out, and this is where I remember this gene, it's linked to so many disorders. And it's like this hot gene that is actually starting to be investigated, although it's not necessarily coming up in the genome-wide, GWAS sequencing studies of thousands of people, but because the effect sizes might be a bit low. But anyway, so we're like, okay, this seems to be very relevant even in the human condition, so we'll further validate this gene. So this is the quantified version of the sequencing data of this gene. So it's downregulated in adolescence after methamphetamine self-administration compared to saline. And then this is, we took another set of animals, and they were all naive, and they did methamphetamine self-administration as I've shown. And this one we did quantitative PCR to measure mRNA levels in a separate sample, but just targeted to this particular gene. And actually what people don't realize is that delta CT levels, actually the higher it is the lower it is. It means it requires more cycles to detect the gene. So we replicated the finding that this gene is downregulated in adolescents that have self-administered meth compared to saline. And then finally, we looked at the protein level. So VMAT1 is the protein product of this gene. And we also replicated that what is saline, it's pooled because adolescents and adults didn't show any differences, but only adolescents that self-administers methamphetamine shows this downregulation. So just to summarize the animal findings so far, onset of methamphetamine use during adolescence in animals leads to escalation of methamphetamine taking when it becomes higher in concentration, leads to stronger methamphetamine-seeking when it is unavailable, but it leads to resistance to extinction of a cue that results in stronger cue-induced relapse, and it also leads to more changes in gene expression in the brain, and reduction of this particular gene may indicate ineffective dopamine recycling in the cytoplasm, which may mean that dopamine signaling levels might be reduced in general due to methamphetamine use. But who cares, right, if this is all in rats? So we wanted to ask, well, now that we've tried to sort of copy and have a model in animals that sort of reflect what happens in humans in terms of behavior, well, can these things, extra things we have observed in animals, can be translated into humans? And all of these things are quite difficult to test ethically right now. But we can at least test some of these things. So we decided to focus on extinction of a methamphetamine-associated cue in meth users, and also the gene expression changes, not in the brain, but at least we can genotype them in the blood, and not in the expression way, but in polymorphism way. So, and we can do this because the protein VMAT is actually extremely highly conserved between rats and humans. Also, further on the relevance, this gene is expressed on locus 8 and is linked with schizophrenia. And to me, it's really interesting because schizophrenia is also a neurodevelopmental disorder. It's very tied to age, very tied to sex as well. And siblings that use meth were more likely to develop schizophrenia, a study has shown. They're very comorbid, or they can be quite comorbid, and they share symptoms and a lot of cognitive deficits as well. So we decide to, we hypothesize that in humans, maybe the earlier the age of onset of methamphetamine use, the greater the difference in this gene polymorphism and resistance to cue extinction in people with meth use disorder. And we got here about maybe 2 years ago. And thank God I had collaborators, especially Susan Russell and Yvonne, and my very good student, Alex, so we worked together to get ethics to study these hypotheses in people and to make sure that our animal model can translate so we can keep using it. And just briefly, I don't really want to talk about everything too much, but sort of what it looks like. We screen them first. We critically interest in the age of onset of meth use. So these are healthy controls and people with not acute meth use, but actually diagnosed with chronic meth use disorder. And they're current users because we collect urine samples to make sure that there's methamphetamine. And yeah, so we collect blood sample for genotyping. And then we do extinction. Oh, there's a huge cockroach here. I wish I could show you. Pick it up. I'm like, keep moving on my left periphery. So cue extinction task, and I'm really proud of my student for coming up with this. Don't me to prove. But we thought about, okay, there are a few cue extinction tasks for methamphetamine users out there, but we don't like them because they show like meth-related, cues like syringes or smoking pipe as pictures and video cues, but that they often don't have a control cue to compare against. They often don't compare to healthy controls' response to these cues. Also, when they do have control cues in many studies of cue extinction psychology, it's like nature. It's so benign. It's as a baseline, it's like I feel like it's extra baseline. It's not normal level of arousal we may experience. Or sometimes they use pornography, and which is too exciting as a control cue. So I was like, what can we come up with that's similar as control cue that a meth user, a healthy control would find it as stimulating as a meth cue for a meth user. I don't know, you know what I mean. So I decided food. I said, Alex, find a bunch of pictures related to food, and he matched with color, matched with contrast, so that explains why we have these food-related cues. So we intermixed these trials and saw and measured their craving levels, but more importantly, objective measure of skin conductive response, so we can measure the sweating to see the arousal. And this is very preliminary, and I can't talk about it too much in detail, but our extinction is looking fantastic. Meth users definitely show more arousal early, but with repeated trials of these cues, they show reduction in arousal, and our healthy controls found both cues are equally stimulating. And with our gene data, which is really exciting, but it's too exciting that I couldn't actually put too much on it. These are all unpublished data. We actually, using chi-square tests, definitely found significant differences in genetic polymorphisms in SLC18A1 in control groups compared to adult onset methamphetamine users compared to adolescent onset methamphetamine users. So I can't really say exactly what, but this is very consistent with our animal work. But what's the real benefit of having an animal model? So we are really happy with our model now. We're going to do lots of things with it to understand humans better because we think that it does translate. But what we can really do with animal model is that we can manipulate and ask causality. Does this gene cause vulnerability in behavior? And I'm very fortunate to have colleagues who share their tools. So CRISPR is all the rage. The reason that CRISPR, the gene editing technique, is quite popular is not because we haven't done gene editing before. We've been doing that for decades. But it's because the specificity of how you can target a single gene is quite easy. So it's an enzyme that cleaves to what we call the guide RNA. And guide RNA can be very specific to a gene. So gene specificity is in short. And the reason that it's a popular tool is that guide RNA is really easy to design. It's so easy to design that even I designed it. So we designed this guide RNA to target SLC18A1 to deliver the, so CRISPR normally what it does is it cuts off that gene that it targets, but we actually deactivated that enzyme that cut it off, but fused that enzyme with a gene activator or gene repressor. So we just want to activate or repress this particular gene with this technology. So we have validated these tools. So this was all in a petri dish, but we are ready to go in humans. So we think this is benefit of animal research because you can ask causality. So the model in my lab is that we want to know human behaviors and we want to model that in animals so we can then look at proteins and RNA, DNA and biology structure. But we also want to test that our rodent behaviors can be also back translated onto humans to make sure it stays relevant. And also in humans, you can collect these biological tissue. And based on that data, we want to manipulate the rodents and see whether the behaviors change. And ultimately, hopefully, then we can help people. So thank you. These are the people who did all the work. Yeah.
What animal or human research links methamphetamine addiction to ADHD?
Well, I guess ADHD drugs are quite amphetamine based, so in that way they're linked. What human research have shown that because the drug that is medicating targets the dopaminergic system, and methamphetamine targets the dopaminergic system, but in animals. There's no real good model, in my opinion, of ADHD in animals. I mean, there are lots of ways to measure their attention, but to make it biologically based so that there are individual differences in attention levels hasn't quite been achieved. So it's a lot like schizophrenia. Pre-pulse inhibition, for example, is fantastic. It translates well into humans. When they show pre-pulse inhibition deficit, animals show it too, and you can study that. But when they say, oh, we inject methamphetamine and they move around a lot, or is that really psychosis? I don't know. So I think animal models are not there yet in terms of ADHD, in my opinion.
And I think the next one is a great one, and I know we've discussed a lot over the last couple of years. Are males and female adolescent rats the same? And does testosterone reduce meth use?
Yeah, that's a really good question. We're testing that now. So in other measures, so my other big specialty, because it's another memory-related disorder that's associated to relapse, is anxiety disorders, and I have a lot more work there. And we definitely see adolescent female, male, massive differences in how they process cues, how they inhibit, how they forget, for example, or even how they learn. Or even the maturation of the brain, like females always a bit earlier than males. But in math, we don't know yet. And we really do want to do even things like that only we can do in animal research, like gonadectomy, get rid of their balls and see what happens. You know, we are doing those things now. We can't do that in humans. Not ethically. Sorry.
Okay. I'll read one more out for you. How do you know that the rat is responding to the drug and not the light?
Oh, because when we take the drug away, it reduces responding to the light. So just the light itself is not enough to keep up that responding. Sometimes it is because they have a pretty, uninteresting life, and so having something happen, even though it's a token, might be exciting. But that light can never sustain a response over a couple of days. It always reduces completely, but drug tend to increase the responding.